Early stages of molecular evolution. We do not know very much about the steps in the origin of protein synthesis, and the steps that led to the linkage of protein synthesis to stored and heritable genetic determinants. Several lines of evidence suggest that in the deep history of life, RNA was probably the first genetic material, rather than DNA. Some RNA structures can function as enzymes. Modern-day ribosomes, for example, depend on specialized chains, known as ribosomal RNA, to form the peptide bonds that link amino acids together during protein synthesis. Presumably, ribosomal RNA and ribosomes evolved from simpler and less refined forms of RNA that had some ability to link amino acids together. RNA molecules can be converted into DNA by two chemical modifications: the removal of a hydroxyl OH group of ribose, which turns it into deoxyribose, and the addition of a methyl group, CH3, to change uracil bases into thymine bases. Today, we find viruses that use several different forms of DNA or RNA as their genetic material. These include single-stranded DNA viruses, double-stranded DNA viruses, single-stranded RNA viruses, and double-stranded RNA viruses. Some researchers theorize that viruses may have played a role in evolutionary transition, leading to replacement of RNA as the stored genomic material of cells. Single-stranded DNA has the advantage of greater stability than RNA sequences, and double-stranded DNA has greater capacity for repair. Since DNA can also base pair with complementary RNA, the double-stranded DNA of cellular chromosomes has both the ability to replicate into two identical copies and also to express its protein coding information by pairing with a complementary RNA sequence, which today is known as messenger RNA. In nucleated cells, the messenger RNA provides a way to synthesize proteins when it travels through nuclear pores into the cytoplasm and binds to ribosomes. That way, a master copy of the DNA remains protected and available in the nucleus and can serve as a template to quickly generate large numbers of a particular protein sequence by producing multiple copies of messenger RNA.